Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation with Dr. Roland Herzog, Editor-in-Chief of Molecular Therapy and Professor of Pediatrics at Indiana University, and Dr. Beverly Davidson, Chief Scientific Strategy Officer and Director of the Raymond G. Perlman Center for Cellular and Molecular Therapeutics at Children's Hospital of Philadelphia, and Dr. Bryan Simpson, Preclinical Team Lead at Lattice Bio. They will discuss an article published in Molecular Therapy by Dr. Simpson and colleagues titled Targeted Long Read Sequencing Captures CRISPR Editing and AAV Integration Outcomes in the Brain. ASGCT is proud to present this episode of the Molecular Therapy Podcast in partnership with our upcoming Advancing Gene and Cell Therapy for Cancer conference on October 16th and 17th in Philadelphia and online. This landmark hybrid event brings together innovative treatment approaches and an unparalleled opportunity to immerse yourself in the latest gene and cell therapy research for cancer. Learn more and register today at asgct.org/advancing. And now, Drs. Roland Herzog, Beverly Davidson, and Bryan Simpson.
Hi, Beth and Bryan, I'm really looking forward to this conversation about your paper, and I'm really excited about this because I think that your paper really touches on so many issues that are critical to the field, and I think there will be a lot of interest in learning more about the topic that you covered. But before we start with that, I would actually like to specifically thank Bev for supporting the journal with so many great manuscripts over the years, original research papers, review articles that have really generated a lot of interest in the community. And so thank you for being such a great supporter of our journal. So I think as far as this particular paper goes, it's again a paper that has really lots of general implications, touches, I think, on three main themes, and you can add to that if I'm missing out on something. So I think first, there is the aspect of gene editing in the CNS. And then second, there's the question of which methodology one should use to properly analyze what happened on the DNA level, on the level of the target sequence. What actually does that sequence exactly look like after gene editing? And lots of methodologies are available, but not all really, do the job properly and give us a clear picture of what is happening. And you're obviously helping to solve this problem. And then the third aspect is about what happens to the genome of the delivery vehicle, which is also a critical question for the safety of gene transfer and gene editing. So I think before we get into those questions, I was hoping that perhaps you can explain a little bit the background of the disease that you're targeting and how these DNA repeats, how they cause disease, and why there is a need for gene editing to try to correct that. If one of you can maybe just give us a little bit of background about that.
Yeah, thank you, Roland. I'm happy to give a little background on the work in our lab to try and repair the repeat in triplet repeat expansion diseases. So more broadly speaking, the repeat expansion diseases encompasses a number of different disorders, some 60 plus and counting. Ones that we work on in my lab include Huntington's disease, which is one of the more common, but others include a form of ALS caused by a repeat expansion in C9orf72, or Fragile X, which is one of the more common neurodevelopmental disorders caused by a repeat expansion disease. And unlike C9orf72 or Fragile X, which affect repeats within non-coding region, Huntington's disease is a repeat expansion in a coding region, similar to two other disorders that we work on in the lab, known as spinocerebellar ataxia type 1 and spinocerebellar ataxia type 2, the latter of which was the focus of Bryan's thesis. And we know that individuals that inherit alleles with normal repeat structures are clinically fine. And as these repeats expand to great, if you inherit a repeat that's greater than 35 or so in one of these alleles, whether it's the allele encoding ataxin 2, which gives rise to spinocerebellar ataxia 2, or the allele encoding Huntington, which gives rise to Huntington's disease. If you inherit the bad allele, you are likely to get the disease. If you do not inherit that allele, you're fine. And so it just stands to reason that if you could chop out that repeat, that you may have benefit. And we know from a host of other preclinical studies that if you can reduce the expression of that disease protein, that it's beneficial in preclinical models and is currently being tested in clinical trials in people. With CRISPR, we thought that we could take a one-shot approach to creating an indel either near the repeat expansion or around the repeat expansion to allow for contraction of the repeat. And that was the focus of Bryan's project. But it became obvious to us that as you create these DNA breaks with an active nuclease, that AAVs have an opportunity to integrate in ways in that fragmented DNA. And so it became very paramount for us to begin to develop methods to look at that.
So you used two approaches to get rid of these repeats, right? Maybe Bryan can explain a little bit, how, what the advantage is one versus the other, and why what the intent was why you comparing those two.
Sure.
Yeah, of course. So we were developing two strategies. One was with a single guide RNA downstream of that CAG repeat to create an indel, and through error-prone DNA repair, knock it out and lower expression of mutant ataxin 2 through nonsense mediated decay. And the second strategy was using two guide RNAs flanking the CAG repeat to excise it. And as Bev said, to really remove that pathogenic repeat, which causes disease.
I see.
We didn't really have any idea, right, which one was going to work.
We did not, no.
Yeah.
So we were exploring both strategies. That's right. Yeah. Yeah. And so, through developing both of those strategies in the lab and then getting into mice and, we're exploring, looking at protein reduction and then looking at the DNA level. And at that time, there was reports of AAV integration coming out in the literature in 2019. Hanlon and colleagues had shown high AAV integration after inducing a double-strand break with AAV-delivered CRISPR. However, they were using PCR-based methods to look at that on-target editing. So we reasoned, if we were to be able to develop or utilize a method that did not rely on PCR in an unbiased way, what do these edits really look like to get at the ground truth?
And I think also for the brain, we wanted to know how often is this happening? And it's very hard to get a sense of that, right? When you're doing PCR.
Right.
And the other thing that I think Bryan's studies really highlighted was how tricky it is to do this work when you're working with a transgenic animal.
Yeah.
So I think that discovery was sort of an aha moment for you.
It was. In a lot of ways, there's these serendipitous moments working through this. Because we had the transgenic models in the lab when I started, and we didn't think beforehand, what are some of the problems of using a transgenic to look at editing, and working through that, we found, when there's multiple copies in the animal,
how many was there? It was quite a few.
So in one of them, in the 127Q, there are 3 copies, and in the 72Q, the back model, there were 6 copies. So you're editing, 3 or 6 copies here, which is very different from a human patient genome. So we also how is that affecting these editing outcomes? In one of the models, we found these very large rearrangements between transgenes, which I did not expect. It was really when I was in the code and looking at these reads, the raw reads, and seeing repeats near each other, and then we hypothesized for what was going on there. It's very unexpected.
I think the other thing that was unexpected was full-length integration of AAV genomes. And and so many little fragments of AAV genomes, which really raises the question of what's going on when these AAVs convert from a single-stranded genome to a double, and then form these concatenamers in the nucleus, because there's seems to be a lot of little pieces floating around, or in some some way or another they're getting fragmented. I think your work really opened up a whole nother series of questions for which some cool studies can follow on.
Yeah, I agree. That was one of the really cool findings, was what is happening when the single strand DNA enters the cell, because so much fragment was going in. And then another hypothesis we had is, after that episome forms, if nicking of that double strand DNA could also somehow influence repair into a double strand break.
So let's talk a little bit about the technology then that you use to analyze these sequences. We've published quite a bit in molecular therapy over the last couple of years about findings on AAV integration, for example, and about the different approaches to performing the DNA sequencing, and other journals publish as well on that topic. And when you, and Beth alluded to that earlier, when you were looking at what people report in terms of integration frequencies, it's all over the place. I mean, there are huge discrepancies from one study to another, and which portion of the vector could actually integrate. You see anything from, it's just tiny fragments, oftentimes just parts of the inverted terminal repeats, which flank the AAV genome. And other reports claim entire genomes integrated that actually express the transgene. So it's really all over the place. And then you find that, perhaps not unexpected, that depending on which methodology you use, you get certain biases. And obviously you believe that your approach is preferable over some others that have previously been used. So maybe you can explain a little bit that the specific methodology you use and why you think it is a better choice.
Well, I think for assessing whether or not you're going to have a full-length genome integration, I think long-read sequencing is really the only way that you can get full-on coverage through the AAV genome. 5 kb is a lot to ask for high-fidelity PCR without crossover of PCR fragments as you're doing the reaction. And we also thought that to get a sense of the amount of fragments relative to the amount of full length and integration, that a PCR-less method could give us a better hint as to what is happening. Mm-hmm. And I mean, as you would expect, it's a lot easier for little pieces to go in than big pieces. So we did find more little pieces than big pieces.
Right.
The overall integration was low, but it was there, right? And meaningful, I think, to a degree. Mm-hmm. Yeah, what do you think?
Yeah, there's quite a bit of method development that went into play to get it to work. It was really based on a paper from Winston Timp's lab, where they're using Cas9 and a ligation adapter ligation method for long read sequencing. I think that where it really shines in the paper is for assessing on-target editing. As far as there's still some blind spots. Mm-hmm. We did not detect translocations that you can detect with PCR-based methods or amplicon sequencing, where you have higher sequencing depth.
Well, I think you worked out some nuances and tricks to get the method to work, and I think it's worth mentioning some of those.
There was some nuances. Yeah, when we first started working with this, it did not work. So we found that you had to one, shear the high molecular weight DNA, and then two, do some size selection, which removed a lot of the small reads, which increased the efficiency of your sequencing. So that was really the key trick for us to reach that high coverage on target in order to really have the power to assess some of these editing outcomes.
And then also you're careful. You were very thoughtful about designing the guides that would pull down the target fragments. And in some cases you tested 4 and they all worked. In other cases, you had to increase those numbers.
That's right. Yeah. So with the one model, the BAC72Q model, it just kind of worked once we worked out some of the kinks. But the second model, the transgenic with 3 copies, we were having quite a bit of problems. And through that, one of the serendipitous findings was we were able to locate the insertion of the transgene in the mouse genome, which was not known, an unexpected finding. Mm-hmm. And once we were able to do that, then we had more sequence to play with to design guide RNAs for that Cas9 enrichment. Right. Yeah. So there were some challenges at first to get it.
Yeah. I mean, it's clearly not a kind of scenario where you can isolate your DNA and pass this on to a genomics core and say, please give me back the sequences. It's not that simple. So I think others can really learn from that experience.
Yeah, I think you really need to enrich it using this Cas9 method. I'd say that's the place where you need to be the most careful, is designing those guides in the right orientations the right directions. And so knowing everything about the flanking sequence is really paramount.
Yeah, because there is some directionality to the design for the sequencing. That's right.
And so then with what you found in the end, does it make you optimistic about the gene editing approach? Where do you think it stands? Does it need to improve more? How much more? And then, of course, where do we stand now with the safety of this approach, given some of your findings with integration of sequences that you probably don't desire in your therapy?
I think it does give us pause in terms of using CRISPR wild-type Cas that retains its active nuclease. I think a nickase is likely fine, that you would, of course, couple to either base editors or prime editors. I think there's just inherently more safety in those approaches. The only other way that I would imagine, that you would feel completely safe with the active nucleases, and we hypothesized this, I believe, in the paper, was that if you could refrain from actually expressing the Cas9 until the genomes were fully assembled as an episomal element at some time post-transduction, you know as well as we do, Roland, that may be months or maybe longer, who knows? And if you can allow that episome assembly to occur where there's just simply too big to integrate into a breakpoint that is going to get closed pretty quickly as that DNA repair machinery comes in, I think that is another way that you could probably use an active nuclease. So you'd want some sort of regulated system that you do your gene transfer, you wait some period of time, and then you induce the nuclease expression. And I think that will work. I definitely think that would work. And that was an experiment that we really wanted to do, but Bryan needed to move on and get a PhD. So.
Oh, no.
We do have a tool to do that, to answer that question.
Yeah.
I think that makes sense to me. And, what presentations I've seen from labs who actually wanted that AAV sequence to integrate for their therapeutic purpose. If you didn't have the Cas activity around that time, it wouldn't work. If you do this too early, too late, so it has to be coordinated. And so if you could separate these two, that would I would imagine that would enhance safety.
Yeah, exactly.
Do you have any evidence whether I understand you explained earlier that the integration of a full vector genome, you did find evidence for that. And it does happen. But the frequency overall is relatively low. So I don't know if you had a chance at all to assess that. I'm curious if you look at, for example, at methylation patterns and so on, do you think it's actually that those genomes are actually expressed after integration?
I don't see why not.
Yeah, it's a great question. We tried some preliminary experiments to address that. And that is part of the reason why I looked at methylation to get at that question. But I agree.
It's such a low frequency. I think the way we would probably go back and look at that is through a fish type experiment where you could actually look at a transcript. Again, it'd be a little hard to know that's coming from an integrated allele versus an episome, but it should be a blob if it's coming from the episome under fish, and it could be, a dot if it's coming from an integrated allele. But it's a difficult experiment. But I really don't. If it's not methylated, which is what Bryan showed, at least out to a month, I don't have any reason to believe that it wouldn't be expressed. It's got the promoter. It's got a poly A.
The question is, does that methylation change over time? So, yeah, we were only taking a snapshot at 4 weeks post-transduction.
I guess AAV vectors tend to some extent, integrate randomly at other places as well, right? Did you have a chance to look at that, how just off-target integration versus at the site where you actually introduced a cut?
This method, by nature, cannot look random. I mean, you would have to tile the entire genome and see if you found it. And here we know where we think it's going to go. And so your Cas9 enrichment happens, in the regions that flank that site. And there's a lot less breaks in neuronal DNA, we believe. Obviously, there's DNA repair going on all the time, but the number of breaks for AAV integration, I think, are not well known in the brain. And so if we happen to know those, I think, it would give us an opportunity to go back in and enrich for those and see.
Yeah, to use maybe a genome-wide approach first, and then come in with a targeted PCR-free method to then really interrogate at those sites.
You're right. That'd be the best way to do it.
those integrations that you see of vector sequences at the site were that you actually targeted, if that versus the events where there is no vector integration, does the vector integration, you believe, interfere with your therapeutic approach? Do you get the desired effect even if there is some portion of the AAV integrated?
Yeah, we never went out that far, Roland. It's a good question. And we were so sort of taken with trying to figure out what was happening post AAV entry. You could still get your desired effect in inserting itty bitty pieces or big pieces into the genome with essentially ablating the expression at that allele. Mm-hmm. The question is, what else are you expressing there that may in and of itself be toxic? Little pieces of AAV, little pieces of Cas that, in our case, we had not reduced CpG dinucleotides in this Cas9 vector or anything like that. So it could be that we'd be expressing more immunogenic problem-causing open reading frames than what were originally there. We just don't know. Mm-hmm.
Yeah, I guess I was, wondering a little bit about risk versus benefit kind of thinking, because there are these risks that you're pointing out and some unknowns. Mm-hmm. But at the same time, you're also trying to treat very devastating diseases. And so, with any drug, any therapeutic approach, there are going to be some side effects, some potential for something else, yet not giving the therapy is obviously, not what the patient would desire. So,
yes, I mean, you raise a really good point, the risk versus benefit here. I think, we can make mountains out of molehills when we find little problems for sure. I think in this case, it was, the amount of on-target editing was good, but it wasn't so great that the small amount of AAV integrations were not concerning and we could just wash our hands and move on. Mm-hmm. I think in this case, it was really kind of a balance of, I think we need another method to either remove these CAG repeats or silence the gene, again, maybe taking advantage of CRISPR-Cas,
right,
or trying different approaches.
Yeah, and in some cases, the AAV integration was as high as the indels at the on-target site. And the question of whether, the AAV integrations, how they're going to disrupt expression of that mutant allele, it's not a question that we directly addressed.
I do think that other studies that have been performed and published, now that folks who've done these can read your paper, might want to consider actually going back and reanalyzing or re-examining their DNA using your method, because they might have clearly missed something or used a methodology that biased them towards certain conclusions that need to be adjusted.
Absolutely agree.
Yeah, I completely agree. I think the field has, at least for gene therapy to the brain, probably, and for many gene therapies based on CRISPR-Cas for dominant disorders, aside from cancer, I think active nucleases may not make sense. That said, the zinc finger approaches, again, those are now being used to direct things to DNA strands and bring in other machinery to silence alleles as opposed to cut them. Mm-hmm. And so I think that Cas is a phenomenal molecule, as are some of the other engineered DNA binding proteins that can target particular sites and bring in the appropriate machinery to either silence or activate the genome, depending on the need.
Is there anything about your study or the topic that I kind of missed with my questions, or, things that you want to add or touch upon that I didn't consider or that we haven't talked about in the conversation so far? If not, if there are, feel free to chime in, but
I think you really hit on the key take-home messages in the beginning with your 3 points there.
I guess
Yeah.
you could add on the whole conundrum of transgenic models when you're working with an active nuclease, and
that is a consideration.
Yeah, and making sure you know the genetics of your model, the inside and out before moving forward.
Mm-hmm. Yeah, I think if we were to go back, we'd want to use a knock-in model.
Yeah,
perhaps
2 alleles is more fun than more.
Well, yeah, it sounds like you did have a lot of fun with that study, but
it was a lot of fun.
But it is certainly
definitely a jigsaw puzzle, right?
It sure was.
Yeah. It shows your ingenuity and your persistence and persevered in the end, and I think taught everybody a lesson. So maybe with that, what I like to do in concluding our discussion is just to learn a little bit about. We know that the famous Dr. Davidson, and I think everybody in our society knows about her accomplishments. So maybe that's an opportunity for Bryan to tell us a little bit about, how you ended up in this fantastic lab and work environment, research environment, how, an institution and where you're at with your career now, and how you see this going forward. Because there are always in these, when we broadcast, these conversations, there are always a lot of trainees who chime in, and they do want to obviously learn about your research and about the paper, but they're also always curious what happens to the people that have done these studies.
Absolutely.
Yeah, of course. So, it's a real
start way back.
Yeah. So it's one, it's a real honor for me to sit here next to my advisor. And so, where I started was in Paul Patterson's lab at Caltech, engineering AAVs with Ben Deverman. He taught me molecular biology at the bench. That's where I learned about Huntington's disease and a lot of these, neurodegenerative diseases, both mechanistic, disease biology, but Paul's lab also was interested in developing therapies. And so that's where I learned about Dr. Bev Davidson and her work using AAVs for gene transfer to the CNS. I knew I wanted to stay in the CNS space. And when I heard that she was being recruited to Penn and I was accepted at Penn for graduate school, I immediately emailed her, and that's where I knew I wanted to do my thesis work. And so, I would say it was a wonderful experience, rigorous training. I had the opportunity to, learn preclinical mouse studies, as well as pick up bioinformatics, learn about gene editing, and continue working with AAVs. And that's what I'm doing now. So Bev's lab spun out a company, Lattice Bio, and I am working there as a scientist and continuing to develop therapies using AAVs in the CNS.
And working on
Huntington's disease. That's right. So it's come full circle from where I was working with Paul. Yeah.
Yeah, that's fantastic. I think it illustrates kind of how this field works, that the gene therapy research, it's so translational, and you have this close connection between basic science and application, and it goes back and forth, and they're so interconnected. So that makes perfect sense. So
right. And there's, still these unknowns about AAVs that, we touch on in this paper.
Mm-hmm.
So, yes.
And, the translational side, I think, is important. So Bryan's exposed to physician scientists. They're moving this to the bedside. And being here in the Center for Cellular and Molecular Therapeutics at CHOP, which I think you probably know well, Roland. We're sitting here in Dr. High's old office looking out at the railroad tracks. And it's really a wonderful opportunity for Bryan to see, things get moving from the preclinical and research vector core level to the clinical vector core and into patients, and all the hard work that happens by so many people, to make that happen. So it's a really exciting time.
Completely agree. So, well, congratulations to both of you again. And thank you not only for the conversation, but of course, mostly for publishing this influential paper with us. So,
and thank you, Roland. This was a real pleasure and an honor.
It was a lot of fun. Yeah.
We were really thrilled to have that email. We were excited, and it's been fun.
Yeah, thanks for the opportunity.